Bismillahirrahmanirrahim. I'm your course instructor of the subject of physiology. My name is Dr. Qurrat Ain Nazar. So today we are going to study about the hemoglobin and iron metabolism. And in this chapter we will cover the basic introduction of the hemoglobin and iron metabolism. Then we will study the normal hemoglobin content, and then we will move towards its functions and the structures. Then we will study the types of normal hemoglobin, abnormal hemoglobin, derivatives, and then we will move towards the synthesis, destruction, and iron metabolism. So let us discuss something about the hemoglobin. As we all know that hemoglobin is the basic essential components of the blood. So hemoglobin is the iron containing coloring matter of red blood cell, and it is a chromoprotein which forms basically 95% of dry weight of RBCs and 30% to 34% of wet weight. So the function of the hemoglobin is to carry the respiratory gases, oxygen and carbon dioxide. It also acts as a buffer system. As we all know that hemoglobin performs a major buffer system role in maintaining the acid-base balance of our body. So the molecular weight of the hemoglobin is round about 68,000. Now let us discuss the normal hemoglobin content. The average hemoglobin HB content in blood is round about 14 to 16 gram per deciliter. However, the value varies depending upon the ages and sexes of the individuals. So if we discuss about the age, at the level of the birth, the amount of hemoglobin content is 25 gram per deciliter. And at third month, the baby achieved 20 gram per liter of the hemoglobin content. And after the 1 year, the baby have a 17 gram per deciliter of hemoglobin content. So from puberty onwards, the content of the hemoglobin in blood is become 14 to 16 gram per deciliter. And at the time of the birth, hemoglobin content is very high because of increased number of RBCs. If we talk about the hemoglobin content in gender, then in adult males, its content is round about 15 gram per deciliter, and in adult females it is about 14.5 gram per deciliter. Now let us discuss the very important functions of the hemoglobin. The first very important function of the hemoglobin is the transport of respiratory gases. And the main function of the hemoglobin is the transport of respiratory gases, and these gases are the oxygen and carbon dioxide. It is responsible for the transportation of oxygen from the lungs to the tissues, and then the carbon dioxide as a result comes out from the tissues to the lungs. So let us discuss the first function, which is the transport of oxygen. When oxygen binds with the hemoglobin, a physiological process which is called the oxygenation occurs, and it results in the formation of the oxyhemoglobin. It means when oxygen combines with the hemoglobin, it forms oxyhemoglobin. The iron remains in the ferrous state in this compound. So the oxyhemoglobin is an unstable compound and the combination is reversible. That is, when more oxygen is available in the body, it combines with hemoglobin, and whenever oxygen is required, hemoglobin can release oxygen reality. So, when oxygen is released from the oxyhemoglobin, it is called reduced hemoglobin or ferrohemoglobin. The second very important function of the hemoglobin, it helps in the transportations of the carbon dioxide. So when carbon dioxide binds with hemoglobin, carb hemoglobin is formed, and it is also an unstable compound, and the combination is reversible. That is, the carbon dioxide can be released from this compound and it can be expelled out from the body. And the affinity of the hemoglobin for carbon dioxide is 20 times more than that of the oxygen. That is why the oxygen, which combines with the hemoglobin and forms oxyhemoglobin, when it distributed in the compartment of the body, it quickly releases from the hemoglobin. But when carbon dioxide attaches with the hemoglobin, its affinity of attachment is more as compared with the oxygen, and carbon dioxide is excreted through the lungs. Buffer action. As we already discussed in detail that how hemoglobin is responsible for maintaining the buffer action in acid-base balance. Yes, it plays a very important role in maintaining the acid-base balance. And this hemoglobin actually maintains the pH of the body. If this hemoglobin alters its reaction, then the pH of the body gets altered. So, we already discussed the role of buffer action of hemoglobin in our previous chapter of acid-base balance. So, we will move towards the next section. Now, if we discuss the structure of the hemoglobin, then hemoglobin is a conjugated protein. It consists of a protein which is combined with an iron-containing pigment. The protein part is actually globulin in nature, and the iron-containing pigment is heme. It means that hemoglobin have two parts. One is the heme, which is iron-containing compound, and the other one is the globin, which is the protein-containing compound. Heme also forms a part of the structure of the myoglobin, and this myoglobin is actually have the ability of the oxygen binding pigment in muscles. And the second one is the neuroglobin. Neuroglobin is actually having a ability of the oxygen binding pigment in brain. It means that these two, myoglobin and neuroglobin, helps in the binding pigment in the muscles and in the brain. Iron. Iron is normally present in ferrous in the form of the Fe2+. So it is unstable and loose form, and in some abnormal conditions the iron is converted into ferric, which is Fe3+ state, which is a stable form. Porphyrin. The pigment part of heme is called the porphyrin. It is formed by 4 pyrrole rings, and these pyrrole rings are called the tetrapyrrole, and these are named as the tetrapyrrole ring 1, 2, 3 and then 4. These pyrrole rings are actually attached to one another by a methane CH4 bridge. The iron is attached to N of each pyrrole ring and N of globulin molecule. Globin. If we look towards the globin, that globin contains 4 polypeptide chains. And among the 4 polypeptide chains, 2 are chains of alpha chains, and then the 2 chains are the beta chains. It means that globin have 4 polypeptide chains. The 2 are the alpha and 2 are the beta chains. This table actually tells you the molecular weight and number of amino acids which is present in the polypeptide chain. And here you can see the molecular weight of the alpha chain, which is about 16,001 to 5. And the amino acids in alpha chain is 141. And if you see the beta chain, the molecular weight of the beta chain is 16,886. And the amino acids in the beta chain is 146. Now let us have a look towards the type of the hemoglobin. If we discuss the type of the hemoglobins, then hemoglobin is of 2 types. The first one is the adult hemoglobin, which is HbA, and the second one is the fetal hemoglobin, which is HbF. Replacement of fetal hemoglobin by adult hemoglobin starts immediately after the birth. It is completed at about 10th month to the 12th month after the delivery. So both the types of the hemoglobin differ from each other structurally and also functionally. Let us understand that what is the structural difference of the hemoglobin. In adult hemoglobin, the globin contains 2 alpha chains and 2 beta chains. And in fetal hemoglobin, there are 2 alpha chains and 2 gamma chains instead of beta chains. Functional differences. And if we look at the functional differences of the hemoglobin, then we came to know that functionally, fetal hemoglobin has more affinity for oxygen than that of the adult hemoglobin. That is the reason that newborn babies have more oxyhemoglobin in them. And the oxyhemoglobin dissociation curve of fetal blood is shifted to the left. Abnormal hemoglobin. Abnormal type of hemoglobins or hemoglobin variations are the pathological mutants form of the hemoglobins. And these variants are produced because of the structural changes in the polypeptide chains. And if there is any structural changes in the polypeptide chains, then it causes the mutations in the genes of the globin chains. So most of the mutations do not produce any serious problem. Occasionally few mutations result in some disorders. And these disorders are called the congenital disorders. So there are 2 categories of abnormal hemoglobin. One is the hemoglobinopathies, and the second is the hemoglobin in thalassemia and related disorders. Hemoglobinopathies. Hemoglobinopathies is a genetic disorder which is caused by the abnormal polypeptide chains of hemoglobin. And some of the hemoglobinopathies are hemoglobin S, hemoglobin C, hemoglobin E, hemoglobin M. Now let us understand the abnormalities of these hemoglobin in detail. In hemoglobin S, it is found in sickle cell anemic patients, and in this the alpha chain are normal. But in sickle cell anemia, the beta chains are the abnormal. It means that hemoglobin has have normal alpha chains but abnormal beta chains. And if you look the hemoglobin C, then the beta chains are actually abnormal in hemoglobin C. And it is found in the people with hemoglobin C disease, which is characterized by mild hemolytic anemia and splenomegaly. Hemoglobin E. Here also the B chains are abnormal, and it is present in people with hemoglobin E disease, which is also characterized by mild hemolytic anemia and splenomegaly. Now let us understand that what is hemoglobin M. It is the normal hemoglobin present in the form of the methemoglobin. So it occurs due to the mutation of genes of both in alpha and beta chains. It means that if alpha and beta chains both have mutations, then it present in the form of the methemoglobin. And it results in abnormal replacement of amino acids. So it is present in babies which are affected by the hemoglobin M, and these babies are called the blue baby syndromes. It is an inherited disease which is characterized by the methemoglobinemia. Hemoglobin in thalassemia and related disorders. If we discuss the thalassemia and related disorders from the point of view of the hemoglobin, then we came to know that in thalassemia, different type of abnormal hemoglobins are present. The polypeptide chains are decreased, and sometimes these chains are absent or abnormal also. In alpha thalassemia, the alpha chains are decreased, absent or abnormal. And in beta thalassemia, the beta chains are decreased and absent, and sometimes they are abnormal. So some of the abnormal hemoglobins are found in thalassemia, and these are the hemoglobin G, hemoglobin H, and sometimes Bart's, Kenya, Lepore, and Constant Springs. Now let us understand something about the abnormal hemoglobin derivatives. There are some hemoglobin derivatives which refer to a blood test to detect and measure the percentage of the abnormal hemoglobin which is actually present in the blood. Hemoglobin is the only carrier for the transport of oxygen. It means that hemoglobin is the life of the body, and without which the tissue death occurs within few minutes. It means that if hemoglobin does not carry oxygen to the tissue of the body, then tissue will become necrot, and the necrosis tissue will ultimately get die. So when hemoglobin is altered in the body, its oxygen carrying capacity is also decreased, which results in the lack of oxygen in the body. So it is important to know about the causes and the effects of the abnormal hemoglobin derivatives in our body. And the abnormal hemoglobin derivatives are formed by carbon monoxide, which is the poisoning, and due to some drugs like nitrates, nitrites, and sulfonamides. Abnormal hemoglobin derivatives are the carboxyhemoglobin, methemoglobin, and the third one is the sulfhemoglobin. And the normal percentage of hemoglobin derivatives in normal hemoglobin is carboxyhemoglobin is round about 3% to 5%. Methemoglobin is round about the less than of 3%, and sulfhemoglobin is in trace. It means that it is undetectable. So if there is any abnormality of high levels of these carboxyhemoglobin, methemoglobin, and sulfhemoglobin, then these high levels actually cause the abnormality in the hemoglobin derivatives in the blood, and they produce some serious effects. So these derivatives actually prevent the transport of oxygen, which results in the oxygen lack in tissues and which may be fatal for the body. Carboxyhemoglobin. Carboxyhemoglobin or carbon monoxyhemoglobin is the abnormal hemoglobin derivative which is formed by the combination of the carbon monoxide with hemoglobin. It means that when hemoglobin combines with the carbon monoxide CO, it makes the carboxyhemoglobin, and actually the carboxyhemoglobin is toxic for the body. Carbon monoxide is a colorless and odorless gas. Since the oxygen has 200 times more affinity for carbon monoxide than oxygen, it hinders the transport of oxygen, results in the tissue hypoxia, and later this tissue hypoxia leads to the death of the tissue. Normally 1 to 3% of hemoglobin is in the form of the carboxyhemoglobin. Now let us see that what are the sources of the carbon monoxide. And these sources are the charcoal burning, coal mines, deep wells, underground drainage system, exhaust of gasoline engines, gases from guns and other weapons, heating system with poor and improper ventilation, smoke from fire, tobacco smoking. Now what are the signs and symptoms of the carbon monoxide poisoning? While breathing air with less than 1% of carbon monoxide, the hemoglobin saturation is 15 to 20% and mild symptoms like headache and nausea appear. The second sign and symptom of carbon monoxide poisoning in the body is while breathing air with more than 1% of the carbon monoxide, the hemoglobin saturation becomes 30 to 40%, and it causes severe symptoms like convulsions, cardiorespiratory arrest, unconsciousness, coma, and sometimes this hemoglobin saturation increases above 50% and it causes the death of the body. Methemoglobin. Methemoglobin is actually the abnormal hemoglobin derivative when iron molecule of hemoglobin is oxidized from normal ferrous state, and this ferrous state is actually converted into the ferric state. Methemoglobin is also called the ferrihemoglobin, and the normal methemoglobin level is 0.6 to 2.5% in our body. It means that this normal methemoglobin, which is 0.6 to 2.5%, is present in the total hemoglobin. And under normal circumstances, body faces the threat of continuous production of methemoglobin. But it is counteracted by erythrocyte protection, and this erythrocyte protective system is called the nicotinamide adenine dinucleotide, which is NADH system, and it operates through the two enzymes. The first one is the diaphorase 1, and it actually indicates the nicotinamide adenine dinucleotide phosphate, which is NADPH, and it is a dependent relactase enzyme. It means it depends for 95% of the action. And the second one is the diaphorase 2, which is the NADPH dependent methemoglobin relactase, and it is responsible for the 5% of the action. And these two enzymes prevent the oxidation of ferrous iron into the ferric iron. Methemoglobinemia. If we discuss the methemoglobinemia, then it is a blood disorder which is characterized by the high levels of methemoglobin in the blood, and it leads to the tissue hypoxia, which causes cyanosis and other symptoms. Now let us discuss some of the causes of the methemoglobinemia. If we see the causes, then methemoglobinemia is caused by a variety of factors, and some of the common factors are present in our daily life, and these are the well water contaminated with nitrates and nitrites. The another factor is the fire, then the laundry ink, matchsticks, explosives, meat preservatives which contains nitrates and nitrites, mothballs, naphthalene balls, rooms, deodorizers, propellants, exposures to the industrial chemicals. And these exposures to the industrial chemicals are the aromatic amines, fluorides, irritant gases like nitrous oxide and nitrogen-zene, and then propylene glycol dinitrite. The third one is the drug, which is antibacterial drugs like sulfonamides, antimalarial drugs like chloroquine, sometimes the antiseptics, and then the inhalants of cyanide antidote kit, and the local anesthesia like benzamides. And there are some hereditary trait factors. Due to the deficiency of the NADH dependent relactase or presence of the abnormal hemoglobin M, hemoglobin M is common in babies, which we already studied that these hemoglobin N actually affect the babies and cause the blue baby syndromes. It is a pathological condition in infants which is characterized by the bluish skin, discoloration, cyanosis, and it causes the congenital heart defect CHD. Now let us discuss that what is self-hemoglobin. If we discuss the self-hemoglobin, that self-hemoglobin is the abnormal hemoglobin derivative, and it is formed by the combination of the hemoglobin with hydrogen sulfide. It is caused by the drugs such as the phenacetin or sulfonamides, and the normal sulfhemoglobin level is less than 1% of the total hemoglobin which is present in our body. And the sulfhemoglobin, if once converted, it cannot be converted back into the normal hemoglobin. So one way to get rid of this from the body is to wait until the affected RBCs completed their normal lifespan with the sulfhemoglobin, and then they automatically destroyed after the process of the normal lifespan of the RBCs. So when new matured RBCs are formed, these previous sulfhemoglobin automatically destroyed and excrete out from the body. Now let us discuss that is there any blood level of sulfhemoglobin in the body? Yes, normally very negligible amount of sulfhemoglobin is present in the blood which is non-detectable. But when its level rises above the 10 gram per deciliter, then the cyanosis appear, which is the bluish discoloration of the skin. It actually indicates the deprived of oxygen in the body, and usually serious toxic effects are not toxic due to the raised level of the sulfhemoglobin in the body. Synthesis of the hemoglobin. If we discuss the synthesis of the hemoglobin actually, then it starts in proerythroblast stage. However, hemoglobin appears in the intermediate normoblastic stage only, which we have already discussed previously when we are studying the proerythroblast chapters. So the production of the hemoglobin is continued until the stage of the reticulocytes formation. And the heme portion of the hemoglobin is synthesized in mitochondria through the process of the profiling and with other coenzymes, which is discussed earlier. So the heme portion of the hemoglobin is synthesized in mitochondria, and the protein part, which is the globin, is actually synthesized in the ribosomes. Now let us discuss the synthesis of the heme portion of the hemoglobin. So the heme is actually synthesized from succinyl-CoA and the glycine. And the sequence of events in the synthesis of hemoglobin is, the first event is the first step is the heme synthesis which takes place in the mitochondria of the cell. Two molecules of succinyl-CoA combines with the two molecules of the glycine and condense to form the S-aminovalinic acid, which is the ALA. And by the synthesis of the X-aminovalinic acid by ALA synthase. And this ALA synthase is further transported into the cytoplasm. When this ALA transported towards the cytoplasm, two molecules of the ALA combine to form a porpholinogen into the presence of the ALA dehydratase. And in the third step, this porpholinogen is converted into uroporpholinogen 1 by the uroporpholinogen 1 synthase. And the fourth step is when this uroporpholinogen 1 is converted into uroporpholinogen 3 by porpholinogen 3 co-synthase. And the fifth step is from when this uroporpholinogen 3, it is converted into a ring structure called copropopyrinogen 3. So this copropopyrinogen 3 is transported back to the mitochondria where it is oxidized to form protoporpyrinogen. And this protopyrinogen is actually carry the factor 9 by the copropopyrinogen oxidase. And in the seventh step, this copropopyrinogen oxide converted into the protopyrin 9 by protopyrinogen oxidase. And in the last step, protopyrin 9 combines with iron to form heme in the presence of the ferrocolloidase. Formation of the globin. As we discussed the formation and the synthesis of the heme, now let us discuss that how globin portion is formed. Polypeptide chain of globin are produced in the ribosomes. There are 4 types of polypeptide chains, and these are named with the alpha, beta, gamma and delta chains. And each of these chains differ from other by the amino acid sequence. So each globin molecule is formed by the combination of 2 pairs of chains, and each chain is made up of 141 to 146 amino acids. And the adult hemoglobin contains 2 alpha chains and 2 beta chains. So fetal hemoglobin contains 2 alpha chains and 2 gamma chains. Now let us discuss the configuration of the hemoglobin. Each polypeptide chain combined with 1 molecule. Thus, after the complete configuration, each hemoglobin molecule combines 4 polypeptide chains and 4 heme molecules. Here you can see the synthesis of the hemoglobin, where we have discussed that the heme portion is synthesized in the mitochondria portion of the cell, and the globin portion is actually synthesized in the cytoplasm. And these steps and the synthesis process are already discussed in detail. So we will move towards the next topic, which is the destruction of the hemoglobin. After the lifespan of 120 days of the RBCs is destroyed in the reticuloendothelial systems, which is particularly designed in the spleen, and the hemoglobin is released into the plasma. Soon the hemoglobin is degraded in the reticuloendothelial cells and splits in the globin and heme. The globin is utilized for the resynthesis of hemoglobin, and heme is degraded further into the iron and porphyrin, where iron is stored in the body as a ferritin and hemosiderin, which are result for the synthesis for the new hemoglobin. It means that these hemosiderin and ferritin helps in the synthesis of the new hemoglobin. And if we discuss about the porphyrin, then a porphyrin is converted into the green pigment, which is called the biliverdin. And in human being, most of the biliverdin is converted into a yellow pigment, which is called the bilirubin. Bilirubin and biliverdin are together called the bile pigments. Iron metabolism. Let us discuss some of the importance of the iron. Iron is an essential mineral and an important component of proteins which is involved in the oxygen transport. So, human body needs iron for the oxygen transport, and iron is important for the formation of the hemoglobin and myoglobin. So, iron is also necessary for the formation of other substances like cytochrome, cytochrome oxidase, peroxidase and catalase. If we discuss the normal value and the distribution of iron in the body, then we have to know the total quantity of iron in the body, which is about 4 grams. Approximate distribution of iron in the body is hemoglobin in the body is 65 to 68%, and the iron in the muscle as myoglobin is 4%. As intracellular oxidative heme compound is 1% in the plasma, so the plasma as a transferrin is 0.1%, and it is stored in the reticuloendothelial system, which is 25 to 30%. Dietary irons. Dietary irons are also available in two forms, which are called the heme and non-heme dietary irons. If we talk about the heme iron, that heme iron is actually present in fish, meat and chicken. And iron in these sources is found in the form of a heme. So heme iron is absorbed easily from the intestine, and non-heme iron in the form of the non-heme iron is available in vegetables, grains and cereals. So the non-heme iron is also not absorbed as easily as the heme iron. Cereals, flours, products of grains which are enriched or fortified. It means that these are much strengthenable than the non-heme iron. So iron become a very good dietary source of the non-heme iron, which is particularly important for the children's and for the women's. Absorption of iron. Iron is actually absorbed mainly from the small intestine. So it is absorbed through the intestinal cells, which is the enterocytes, by pinocytosis and transported into the blood. So bile is essential for the absorption of the iron, and iron is also present mostly in the ferric form, which is the Fe3+ form. So it is converted into the ferrocene, which is Fe+2, which is absorbable into the blood. And the hydrochloric acid, which is produced from the gastric juices, it makes the ferrous iron soluble so that it could be converted into the ferric iron by the enzymes ferric reductase from enterocytes. So from enterocytes, ferric iron is transported into the blood by a protein called ferroprotein. And in the blood, ferric iron is converted into the ferrous iron and transported. Transport of iron. If we discuss the transport of iron, then immediately after the absorption into the blood, iron combines with a beta globulin, which is called the apotransferrin, and it is secreted by the liver through the bile. So it results in the formation of the transferrin, and iron is further transported in blood in the form of the transferrin. So iron normally combines loosely with the globins and can be released easily by any region of the body. If we talk about the storage of iron, then storage of iron is stored in large quantities in the radiculoendothelial cells, and it is also stored in liver hepatocytes. So in other cells, it is also stored in small quantities, and in the cytoplasm of the cell, iron is basically stored as ferritin in large amount. So the small quantity of iron is also present and stored in the form of the hemodesirin. Daily loss of iron. In males, round about 1 gram of iron is excreted every day through the feces. And in females, the daily loss of amount of iron is very much as compared to the males. This is because of the menstruation. So the 1 gram of hemoglobin contains round about 3.34 mg of iron. So normally 100 ml of blood contains 15 gram of hemoglobin and about 50 gram of iron, which is 3.34 into 15. So if 100 ml of blood is lost from the body, there is a loss of the about 50 mg of iron. And in females, during every menstrual cycle, about 50 ml of blood is lost, by which 25 mg of iron is also lost. So this is why the iron content is always less in female as compared with the males. And iron loss during hemorrhage and blood donation is very important, and sometimes women need this blood donation. So if 450 ml of blood is donated, about 2 to 5 mg of iron is lost. Regulation of total iron in the body. Absorption and excretion of iron are normally maintained equally under normal physiological conditions. And when the iron storage is saturated in the body, it automatically reduces the further absorption of iron from the gastrointestinal tract by the feed-by mechanisms. And the factors which reduces the absorption of the iron are the stoppage of apotransferrin formation in the liver, so the iron cannot be absorbed from the intestines. And the second factor is the reduction in the release of iron from the transferrin, so that the transferrin is sometimes completely saturated with the iron and further absorption is prevented. So this is all about of today's topic of the hemoglobin and iron mechanism. Thank you.